frequently more or less deflected from the median plane, thus lessening the size of one nasal cavity and increasing that of the other. Ridges or spurs of bone growing into one or other cavity from the septum are also sometimes present. Immediately over the incisive canal at the lower edge of the cartilage of the septum, a depression, the nasopalatine recess, is seen. In the septum, close to this recess, a minute orifice may be discerned. It leads backward into a blind pouch, the rudimentary vomeronasal organ of Jacobson, which is supported by a strip of cartilage, the vomeronasal cartilage. This organ is well developed in many of the lower animals, where it apparently plays a part in the sense of smell, since it is supplied by twigs of the olfactory nerve and lined by epithelium similar to that in the olfactory region of the nose. The roof of the nasal cavity is narrow from side to side, except at its posterior part, and may be divided, from behind forward, into sphenoidal, ethmoidal, and frontonasal parts, after the bones which form it. The floor is concave from side to side, and almost horizontal antero-posteriorly. Its anterior 3/4 are formed by the palatine process of the maxilla, its posterior fourth by the horizontal process of the palatine bone. In its anteromedial part, directly over the incisive foramen, a small depression, the nasopalatine recess, is sometimes seen. It points downward and forward, and occupies the position of a canal which connected the nasal with the buccal cavity in early fetal life. The mucous membrane, membrana mucosa nasi. The nasal mucous membrane lines the nasal cavities, and is intimately adherent to the periosteum or perichondrium. It is continuous with the skin through the nares, and with the mucous membrane of the nasal part of the pharynx through the choanate. From the nasal cavity its continuity with the conjunctiva may be traced, through the nasolacrimal and lacrimal ducts, and with the frontal, ethmoidal, sphenoidal, and maxillary sinuses through the several openings in the meatuses. The mucous membrane is thickest and most vascular over the nasal conchae. It is also thick over the septum, but it is very thin in the meatuses on the floor of the nasal cavities and in the various sinuses. Owing to the thickness of the greater part of this membrane, the nasal cavities are much narrower, and the middle and inferior nasal conchae appear larger and more prominent than in the skeleton. Also the various apertures communicating with the meatuses are considerably narrowed. Structure of the mucous membrane. The epithelium covering the mucous membrane differs in its character according to the functions of the part of the nose in which it is found. In the respiratory region it is columnar and ciliated. Interspersed among the columnar cells are goblet or mucin cells, while between their bases are found similar pyramidal cells. Beneath the epithelium and its basement membrane is a fibrous layer infiltrated with lymph corpuscles, so as to form in many parts a diffuse adenoid tissue, and under this a nearly continuous layer of small and larger glands, some mucous and some serous, the ducts of which open upon the surface. In the olfactory region, the mucous membrane is yellowish in color, and the epithelial cells are columnar and non-ciliated. They are of two kinds, supporting cells and olfactory cells. The supporting cells contain oval nuclei, which are situated in the deeper parts of the cells and constitute the zone of oval nuclei. The superficial part of each cell is columnar and contains granules of yellow pigment, while its deep part is prolonged as a delicate process which ramifies and communicates with similar processes from neighboring cells, so as to form a network in the mucous membrane. Lying between the deep processes of the supporting cells are a number of bipolar nerve cells, the olfactory cells, each consisting of a small amount of granular protoplasm with a large spherical nucleus, and possessing two processes, a superficial one which runs between the columnar epithelial cells and projects on the surface of the mucous membrane as a fine hair-like process, the olfactory hair. The other or deep process runs inward, is frequently beaded, and is continued as the axon of an olfactory nerve fiber. Beneath the epithelium, and extending through the thickness of the mucous membrane, is a layer of tubular, often branched glands, the glands of Bowman, identical in structure with serous glands. The epithelial cells of the nose, fauces, and respiratory passages play an important part in the maintenance of an equable temperature, by the moisture with which they keep the surface always slightly lubricated. Vessels and nerves. The arteries of the nasal cavities are the anterior and posterior ethmoidal branches of the ophthalmic, which supply the ethmoidal cells, frontal sinuses, and roof of the nose. The sphenopalatine branch of the inferior maxillary, which supplies the mucous membrane covering the conchae, the meatuses and septum. The septal branch of the superior labial of the external maxillary, the infraorbital and alveolar branches of the internal maxillary, which supply the lining membrane of the maxillary sinus, and the pharyngeal branch of the same artery, distributed to the sphenoidal sinus. The ramifications of these vessels form a close plexiform network beneath and in the substance of the mucous membrane. The veins form a close cavernous plexus beneath the mucous membrane. This plexus is especially well marked over the lower part of the septum and over the middle and inferior conchae. Some of the veins open into the sphenopalatine vein, others join the anterior facial vein, some accompany the ethmoidal arteries and end in the ophthalmic veins, and lastly, a few communicate with the veins on the orbital surface of the frontal lobe of the brain, through the foramina in the cribriform plate of the ethmoid bone. When the foramen cecum is patent, it transmits a vein to the superior sagittal sinus. The lymphatics have already been described, page 695. The nerves of ordinary sensation are the nasociliary branch of the ophthalmic, filaments from the anterior alveolar branch of the maxillary, the nerve of the pterygoid canal, the nasopalatine, the anterior palatine, and nasal branches of the sphenopalatine ganglion. The nasociliary branch of the ophthalmic distributes filaments to the forepart of the septum and lateral wall of the nasal cavity. Filaments from the anterior alveolar nerve supply the inferior meatus and inferior concha. The nerve of the pterygoid canal supplies the upper and back part of the septum and superior concha, and the upper nasal branches from the sphenopalatine ganglion have a similar distribution. The nasopalatine nerve supplies the middle of the septum. The anterior palatine nerve supplies the lower nasal branches to the middle and inferior conchae. The olfactory, the special nerve of the sense of smell, is distributed to the olfactory region. Its fibers arise from the bipolar olfactory cells and are destitute of medullary sheaths. They unite in fasciculi, which form a plexus beneath the mucous membrane, and then ascend in grooves or canals in the ethmoid bone. They pass into the skull through the foramina in the cribriform plate of the ethmoid, and enter the undersurface of the olfactory bulb, in which they ramify and form synapses with the dendrites of the mitral cells. The accessory sinuses of the nose. Sinus paranasales. The accessory sinuses or air cells of the nose are the frontal, ethmoidal, sphenoidal, and maxillary. They vary in size and form in different individuals, and are lined by ciliated mucous membrane directly continuous with that of the nasal cavities. The frontal sinuses, sinus frontalis, situated behind the superciliary arches, are rarely symmetrical, and the septum between them frequently deviates to one or other side of the middle line. Their average measurements are as follows: height 3 cm, breadth 2.5 cm, depth from before backward 2.5 cm. Each opens into the anterior part of the corresponding middle meatus of the nose through the frontonasal duct, which traverses the anterior part of the labyrinth of the ethmoid. Absent at birth, they are generally fairly well developed between the 7th and 8th years, but only reach their full size after puberty. The ethmoidal air cells, cellulae ethmoidales, consist of numerous thin-walled cavities situated in the ethmoidal labyrinth and completed by the frontal, maxilla, lacrimal, sphenoidal, and palatine. They lie between the upper parts of the nasal cavities and the orbits, and are separated from these cavities by thin, bony laminae. On either side they are arranged in 3 groups, anterior, middle, and posterior. The anterior and middle groups open into the middle meatus of the nose, the former by way of the infundibulum, the latter on or above the bulla ethmoidalis. The posterior cells open into the superior meatus under cover of the superior nasal concha. Sometimes one or more opens into the sphenoidal sinus. The ethmoidal cells begin to develop during fetal life. The sphenoidal sinuses, sinus sphenoidalis, contained within the body of the sphenoid, vary in size and shape, owing to the lateral displacement of the intervening septum They are rarely symmetrical.